Hi, everyone. Welcome. If you're new here, I'm Erin Harris, host of Cell and Gene, the podcast. If you're a longtime listener, welcome back. I am genuinely excited to share episode 112 with you. I had the opportunity to sit down with Michael Quigley, Ph.D., who's just wrapped up his first year as Chief Scientific Officer and Global Head of Research at Sanofi. Mike brings a seriously impressive background to his role at Sanofi that spans big pharma, biotech startup, cutting edge science, everything from leading immunology, oncology, neurology, and rare disease research. He's also held senior roles at Gilead, BMS, MedImmune, and Janssen, just to name a few. And with that kind of experience, you'll see why in just a second Mike has a unique 360-degree view on what it really takes to move breakthrough medicines from early discovery all the way to the clinic. Mike and I talk R&D strategy, the future of genomic medicine, and what makes a blockbuster innovation in this space, and how Sanofi is doubling down on both scientific curiosity and collaboration. I could have talked to Mike for hours. He's a wealth of knowledge and is happy to share his insight. And that's why I'm really excited to share this episode with you. And so, with that, here is my conversation with Sanofi's Mike Quigley. Enjoy. Mike, thanks so much for your time. Welcome to Cell and Gene. I'm happy you're here.
Thanks, Erin. Great to be here. Really looking forward to the conversation.
Yeah, me too. So you're new to Sanofi. You're coming up on your year anniversary as Chief Scientific Officer. So congratulations on that. And I'd love for you to talk to Cell and Gene, the podcast audience, about what your top priorities are for shaping the R&D strategy for the company, and also how you see specifically genomic medicines, including cell and gene therapies, fitting into that overall R&D strategy and vision.
Absolutely. Thanks, Erin. It's been a real privilege to have joined Sanofi, as you said, a little over or up to a year ago. Really, my goal as we think about the R&D strategy within Sanofi is to capitalize on the deep expertise and breadth of knowledge we have in immunology, but span the sort of applicability of that as we think about unmet need and diseases with a lens of immunoscience. That is, take the core sort of understanding of key nodes or immunologic pathways and how they pertain to diseases both within the inflammatory or inflammation and immunology space, but more broadly as we think about sort of areas such as neurodegeneration, ophthalmology, diseases that clearly have an immunologic component in terms of driving the disease pathophysiology, but for which our expertise in immunology allows us to sort of extend that knowledge and sort of try to drive toward medicines that fit that unmet need. As we think about transformation and the potential thereof, genomic medicines clearly and squarely sits within that space. We see that transformative potential from a gene therapy perspective for patients, and are clearly advancing these efforts both internally and in partnership with the external community through our dedicated Genomic Medicines Unit that we've built coming up on about 5 years ago within Sanofi. Our focus in the GMU, as we call it, is really around in vivo approaches to gene therapy. And that's really because we see that as a more favorable patient journey, and it decreases the burden on logistics and sort of scales, of which I'm sure we're going to talk about a bit as a sort of lagging issue associated with gene therapy in general. They're part of the modality mix. So as I sort of start to think about distribution across our therapeutic areas, gene therapies play a key role in many of those therapeutic areas as we think about the modality mix within our current portfolio and ones of the future. And because we see the promise of that potential and the challenges, we took a purposeful approach as we thought about building out the GMU to advance gene therapies in the clinic in select cases. And our strategy is informed by near and longer term clinical readouts. So proof points we see in the applicability of gene therapies in the clinic and how that might inform a refinement, if you will, of our portfolio strategy, and particularly as we think about genetic therapies. And sort of lastly, I'd say in the context of how we built that GMU, it's really with a modular component. So there's an in vivo approach and a modular sort of gene therapy platform to address both rare diseases, which we're in today at Sanofi with the Genzyme legacy, but also more common diseases in our other therapeutic areas.
Good, great. And thanks for that explanation. And I want to talk a little bit more about, in fact, the Genomic Medicine Unit, or GMU, as you've been thoughtfully describing to us. How do you plan to build on the efforts to accelerate innovation in cell and gene therapy, but while maintaining and building scalability options and access for the patients globally, not just in the US and then certainly abroad?
It's a key point. So, as I just referenced, our genomic medicine strategy is aligned well across all of our therapeutic area strategies. And we're doing it with a sort of approach, meaning pursuing the portfolio with both an internal organic sort of build as well as a partnering lens as we think about bringing in technologies or developing technologies that solve for some of the both the need and the issues that you're calling out. So we're using both viral AAV-based in vivo approach as well as non-viral based deliveries. And while there have been some challenges based on translatability from preclinical to clinical setting, in particular as we think about sort of immunogenicity risk associated with some of these platforms, we're working on and we see clear solutions and a path forward for many of these. So we're in solutions mode as we think about some of the limitations of prior iterations of sort of delivery of gene therapies. And as you talk about market access and sort of scalability, maybe scalability first, as we built out the GMU, we were very purposeful rather, of co-locating our CMC teams with our research teams. So that process optimization, which ultimately will lead to improved efficiency and scalability, is inherent to the sort of ethos of building out the GMU. And obviously, the goal is not to sort of have this sort of potential transformative, capabilities of gene therapy specific to any particular geography. So as we think about scaling, not only from a process perspective, from a geographic perspective for broader patient need, it's really going to be clarified based on market access routes, effective reimbursement schemes, of course, and our ability to keep lowering costs. So that's where we think they go hand in hand, both this sort of scalability, cost of goods, which ultimately would lead to sort of a broader disbursement from a global footprint perspective. The focus on in vivo approaches that I referenced as we built out the GMU as opposed to ex vivo, we really think enables that or is enabling with respect to sort of that scalability and the patient journey that I sort of referenced with respect to sort of economies of scale and process improvements.
Gotcha. Okay. I want to shift our conversation a little bit and talk about the upcoming WMIF or World Medical Innovation Forum conference that's coming up. By the time this podcast episode airs, the WMIF conference coming up in Boston will have already happened. That said, I have attended WMIF in the past. I've been a moderator there. Excellent, excellent conference to anyone who is planning to attend or is looking to attend. The team behind that conference is wonderful. So, have you already attended a WMIF in the past?
I've not. I'm looking forward to the conference next week.
Yeah, you're in for a treat. So, you're on the panel creating the next blockbuster, excuse me, in I&I. So, as I said, this will have already occurred by the time this podcast airs, but talk to us a little bit about what that actually means, in blockbuster in immunology and inflammation today, especially given the rapidly evolving landscape of advanced therapies. What does that actually look like?
Yeah, Aaron, I'm really looking forward to that panel discussion, and maybe I'll take a broader definition of blockbuster. So there's clearly a commercial context as we think about the common use of the term blockbusters. We think about sales of any particular asset. But my goal really within Sanofi is to start to think about large areas of unmet need, in particular as we think about inflammatory immunologic diseases. We at Sanofi are building upon the sort of broad portfolio we have that targets different aspects of diseases, both within dermatology and respiratory, type 2 diseases in particular, in that space, but broad into areas of rheumatology and gastroenterology. And there's clear unmet need across all those therapeutic areas within I&I diseases. For me, what blockbuster really means is the identification of key nodes or key targets within immunology that have broad applicability over large areas or vast amounts of those sort of diseases or areas. And therefore, the blockbuster potential is to bring unmet need to a large number of patients. We've sort of seen that through blockade of IL-4 receptor alpha with tupilumab, as we think about the impact that's had across a large number of diseases within the type 2 inflammatory space. And so my goal going forward within the Sanofi portfolio, and more broadly within the I&I sort of therapeutic area, is to start to think about key nodes that should complement, for instance, IL-4 receptor alpha in terms of the scale and the applicability to diseases. And as we're thinking about sort of genomic medicine or sort of the approaches thereof and our efforts coming out of the GMU as it pertains to this blockbuster, one area of particular interest and we think of high potential as we think about blockbuster is things like deep B cell depletion or immune reset within the immune system. There's been some really seminal work coming out of George Jett's lab and others as well, showing that using, for instance, CAR T or engagers that deplete B cells within the tissues of autoimmune patients can lead to long-term durable treatment-free remission in diseases that, for instance, were sort of managed much more based on comorbidities as opposed to anything resembling long-term treatment-free remission. So things like lupus, myositis, and the like. And so our efforts within the Genomic Medicine Unit are focused in part on sort of understanding whether this in vivo reprogramming that we talked about a bit earlier could have applicability in the context of deep B cell depletion in autoimmune disease. And I think from a blockbuster potential, really the unmet need and sort of the value to the patient of treatment-free, deep remission of their diseases is really meaningful and blockbuster in the potential. So we're looking forward to sort of seeing that play out both within our own portfolio, but within the field as we're moving in that direction.
Good, good. Okay. And to our listeners who happen to be attending WIMF, be sure to get some information after the fact through the organization based for the session that Mike is going to be on, because I think it's going to be a really good one. That's with all of that said, we know that mediated diseases are very complex. So how do you see cell and gene therapies transforming the way we approach treatment for these conditions compared to traditional biologics or even small molecules?
Thanks, Aaron. I think the promise of sort of gene therapies and the future and where we see the potential is very much complementary to the current approaches associated, as you'd articulate, with biologics or small molecules. We have a sort of an approach that capitalizes on all three sort of classes of modalities, thinking about oral combinations for patients as well as biologic combinations. And really, as you point out, the complexity associated with immunologic or inflammatory diseases, and one is one that we believe really requires sort of combination approaches. And as we think about the modalities that would sort of lend themselves to combination and or the other unmet need from a durability perspective, we think gene therapy really has the potential to think about how to not only target critical nodes that we know are drivers within immune-mediated diseases, but also that durability component and the patient convenience on top of that. So as we think about a future of sort of gene therapy within the I&I space, it's really the potential perhaps to use cells or tissues within the body and turn them into biofactories as we think about sort of durable, sort of continuous treatment that removes the need for repeat administration from some of those other modalities that we currently have available to us and helps us access sort of hard to reach compartments rather within the body that might be sort of sites of disease. And that's really a lot of the efforts we have around sort of targeted delivery, as well as tissue penetration as we think about sort of gene therapies. And maybe going forward, the complementarity of various modalities, for instance, small molecules that activate gene therapies, is not too far off in the future. And that's certainly an area that we're of interest both internally and through partners.
Good, good. Okay. We'll get into specifically partnerships in a little bit, but before we do, given that you are the CSO and uh, global head of regulatory, or excuse me, research R&D, I'd be remiss not to ask you your thoughts on how AI is transforming the space. And so, from your perspective, how is Sanofi in fact integrating AI into your research strategy? Do you have what are the greatest benefits you see coming down the pike? Are you concerned about anything? Where do you really see AI playing a role in the sector overall, but also what Sanofi's working on?
We're all in on AI at Sanofi, and
Yeah.
that permeates all components of the entirety of the R&D organization, but more broadly, the entirety of all Sanofi. And I think in terms of today, where AI is and making the biggest impact as we think about our research portfolio, for instance, is really around the design, make, test, analyze cycle associated with the optimization of any of our molecules as they make their way through the pipeline. So that's one area. The second area that we're really excited by is sort of a lab AI agent, which aggregates both the entirety of the external literature, for instance, in any body of work, and integrates with all of the internal data sets that we have within Sanofi, and allows, with an overlaying large language model approach, allows our scientists to query the entirety of that in a way that, similar to a chat GPT interaction, would propose the next set of experiments and really elevating the science. And most importantly, for both aspects, AI for us within Sanofi is both enabling from a scientific perspective, but also a significant increase in efficiency as we think about how to best sort of progress our portfolio with the ultimate goal of getting medicines to patients faster, of course. As we think about gene and cell therapy, and gene therapy in particular, is internally, AI, consistent with the design, make, test, analyze sort of reference, is used day in, day out as we think about optimization of our molecules. Um, Genomic medicines, as you're well aware, programmable, modular, consisting of delivery vehicles like lipid nanoparticles or viral capsids and payloads, of course, mRNA, DNA, promoters, et cetera. And so this modular nature really creates, in our mind, the ideal environment for advanced algorithms and machine learning, given the LLMs are particularly well suited for innovating on the genetic code, which essentially is another natural language, of course. And so advanced algorithms can improve existing genomic medicines, allowing us to examine more combinations back to where we think we need to go outside of monogenic diseases as disease drivers. And that, this is much more that can possibly be done today in a wet lab. And so we really need that AI ML component to really accelerate the efficiency of the optimization and fundamentally enabling sort of new ways of building genomic medicines. And for us, day in, day out, AI has been deployed to develop new improved AAV capsids, create novel optimized payloads, optimize codon usage in mRNA, novel promoters, and across research labs, really with the goal of, as we've already talked about, improving productivity and efficiency, and ultimately optimization of our molecules.
Good. Okay. And I want to talk a little bit about partnerships, but and the partnerships you value and why between academia, industry, regulators, that type of thing. And perhaps we can incorporate AI into that discussion. And so, as you're building partnerships, certainly around genomic medicines, what do those partnerships look like, given how all in Sanofi is on AI? Is that part of the equation for the partnerships you're looking at? And then, what kinds of partnerships or, that ecosystem building effort do you find most critical for, to move your work forward?
Erin, it's critical that we partner. We don't have a not invented here status, and we think innovation obviously comes from everywhere. And that's both internal efforts as well as sort of seamlessly integrating with the external ecosystem and the environment, and making sure we're at the sort of bleeding edge of understanding where the innovation is going, as we're talking here today in the cell and gene therapy space. So I would bucket it in three main areas: academia, industry, and then regulatory sort of interactions. I think with academia, that's really, where the envelope's being pushed as we think about the next generation of innovation, going beyond what we can currently do alone. So we have sort of sponsor research agreement approaches, and sort of we're well connected within our GMU organization to the external academic community in the gene therapy space to understand what new solutions might be being brought in the broader ecosystem to solve for things like we've already talked about: scalability, optimization, delivery of different payloads that might unlock new mechanisms of actions as we think about gene therapy. So we're quite strong and continue to look for key academic collaborations within the cell and gene therapy space. On industry, there are enabling technologies that are being built within biotechs, for instance, that really we see in a puzzle piece fitting lens that might complement what we already have within our own armamentarium in the cell and gene therapy space. So, those particular areas of innovation, technologies that might either allow us to scale or accelerate what we believe are promising innovations already within our own portfolio. And importantly, the lens is not obviously on the early side or earlier side of the value chain, like academic collaborations, but ones that really accelerate to clinical utility of gene therapy. And that's really that sweet spot for how we look to partner with, for instance, biotechs, both locally within the Boston-Cambridge system, but of course, as a global organization that spans the globe as we think about where that innovation can come from.
Mhm.
And from a collaboration and interaction on the regulatory side, it's really that engagement's critical for us as we think about the conversation with regulators and clear IND approval criteria, which is a bit distinct from maybe the well-trodden small molecule and biologics, or at least open for discussion, perhaps more so. and the conversation with governments to improve clarity on actionable reimbursement schemes. So we talked a little bit about distribution of cell and gene therapy, ultimately globally, hopefully. That's really going to require that close interaction as we think about not only the regulatory process, but in a success scenario of approval, how do we think about really reviewing sort of the reimbursement schemes to make sure that these promising modalities can make it to as many patients as possible.
Great. Okay. Mike, before I let you go, I want to talk a little bit about your thoughts on the next handful of years, 3 to 5 years, and what scientific or technological breakthroughs do you believe are most significantly might change the trajectory of immunology for cell and gene therapy?
Erin, it's the crystal ball question, and one that we're incredibly excited by at Sanofi and within our Genomic Medicines unit. I think maybe a couple of known issues that we believe are solvable, and we believe are solvable ideally in that next 3 to 5 year time horizon. And that's overcoming pre-existing immunity to enable viral gene therapies for more patients. So pre-existing neutralizing antibodies, obviously a hurdle associated with, for instance, AAV-based delivery. And so a solution to bring to bear to that, either on the capsid side and/or on a combination approach as we think about immunologic agents, we're quite excited by. Foundational platforms are solving for a foundational platform to unlock delivery to particular organs, so sort of selective delivery or tropism as we think about particular tissues that might be the areas of disease and which you want to selectively deliver genomic or genetic medicines. That becomes important to us. There's already been sort of a lot of progress in that space. We think unlocking sort of additional tissue tropism or delivery mechanisms with a minimal tolerability issue associated with that is on the horizon as well, and we're quite excited by that. We talked about productivity to increase treatment availability across the globe. We think there's going to be, certainly in the non-viral space, some significant efforts in that space that we're quite close to and excited by in that time horizon. We touched on it a little bit earlier today, but regulatable or reversible gene therapies, as we think about both the safety and activity, tissue specific or selective sort of impact, certainly of interest to us. And maybe lastly, and what we're sort of quite excited by, and there's sort of evolution in the field, we'll see that play out as it ultimately transitions into clinical utility, is really hopefully and ideally a step change in our ability to edit genes at a single nucleotide resolution. So potentially advancing the sort of platform approaches for genetically defined rare diseases, of which Sanofi's committed to based on, again, we talked about our legacy and our go forward sort of ambition to bring benefit to patients with rare diseases. And we think gene therapies in particular are obviously well suited in that space. So we think the future is bright. We think that there's a lot of opportunity within cell and gene therapy. We think our Genomic Medicine Unit is well positioned to sample the innovation both internally as we think about bringing benefit to patients, but equally importantly, as we talked about from the external ecosystem and where that innovation is obviously progressing at a pace much more than what we could do internally. And it's that partnering, that sort of collaborative and collegial mindset that we have within our genomic medicine strategy that we think will ultimately win the day here for patients.
All right, listeners, that wraps up this episode of Cell and Gene, the podcast featuring Sanofi's Mike Quigley. Mike, thanks so much for your time and the insight you shared with us today. This has been a thoughtful and insightful episode.
Thanks, Erin. I really appreciate the time and looking forward to the panel and where gene therapies take us.
Listeners, please visit us in Cell and Gene and subscribe to Cell and Gene, the podcast, so you never miss an episode just like this one. And we'll talk to you soon.